All right, welcome everybody. My name is Dustin Maloney. I'm a HawkWatch International Research Associate, and I'm also a graduate student at Utah State University in Dr. Clark Rushing's Population Ecology Lab. And today I'll be talking to you guys about using raptors as indicator species. All right, and with that, just a quick overview of the presentation today. I've broken it down into 3 different parts. First part, we'll talk about what an indicator species is, how we select them, why they're important. And then I'll go into part 2, talking about kind of a historical case of using raptors as indicator species. And then kind of some of the current and potential future issues that raptors might face and might be good use of for indicator species. And then for part 3, we'll talk about the current research that's going on at HawkWatch International with this topic. And we'll mainly focus on my research. HawkWatch is doing a lot of amazing things in this realm. And for time's sake, we'll kind of keep it just to my part. And then in between each part of these topics, I'll pause for any sort of questions. So if you have questions, feel free to put it into the chat, or you can feel free to unmute during that pause, and I can answer any questions. It's a lot of content here, so feel free to ask questions as much as you like. All right, let's begin. So what is an indicator species? An indicator species is essentially an organism or a group of organisms that can provide some insight about the environment with their health or their population health. So I think kind of the classical example would be the canary in a coal mine, right? So humans are worried about the environmental health, those noxious gases in the coal mines. And so they would bring in a canary that was also susceptible to the same noxious gases to indicate when those gases filled up in the mine. And when that canary died or got sick from those gases, it would give an indication, or it would provide some information to those miners about the health of that environment. So again, an organism or a group of different organisms that can provide some information about that ecological health of the environment. And just kind of reiterate what the indicator species are. This is another great example with bugs. That photo on the left is a caddisfly, and if you're an angler, I'm sure you're familiar with that species. They are very intolerant of pollution. You will not find them in polluted waters. And so, that provides some indication if you go out and do some sampling, like those folks on the right there. If you dig around in the muck and turn over some rocks and you find some bugs, if you don't find any of these caddisflies, it's likely meaning that that water body or that aquatic system has some pollution in it. And actually an interesting thing about this macroinvertebrate survey stuff is that there's a group of organisms, a group of different types of bugs that have different tolerances to pollution. So you can actually kind of gauge how polluted the water is by depending on what species you find within that water body. So again, these are just indicators of the health of that ecosystem. And with these caddisflies, you can base it off the population. If there's no population of them, it's probably polluted. There might be some effluent from anthropogenic sources upstream, or some ranching runoff, nitrates, things like that. But their population is definitely providing you some insight into the pollution of that water body. So that's kind of what an indicator species is, just giving us that ecological health information. So why are indicator species useful? Well, they're good, like I said, at monitoring environmental changes. With that population or their health, they can provide a lot of that information. They're also great at providing some integrity and restoration information. So if you're a land manager and you go out to an area that has been disturbed with invasive grasses, say with cheatgrass, and you want to reestablish the native habitat, say put in some shrub cover, native grasses, you go out there, till the land, put in the shrubs, those native grasses. You might use, say, a raptor, a hawk, or an eagle as an indicator species, because they'll only come back and establish likely when their prey base, those herbivores, have reestablished in that system. And if those herbivores have reestablished in that system, it means that their forage base has also been established. And so you know that that ecosystem has come back together and provided some health to the entire system. So again, in this situation, raptors would be a good option for that kind of land manager to use as a indicator species. It's also very cheap and reliable. The environment is a very complex system. That's like the understatement of the year, right? There's biotic and abiotic components to the ecosystem. So those abiotic components might be the soil, the air, the water, and then the biotic components might be the bugs, the birds, the mammals that live in that system. And to try to sample and test each one of those components would be really expensive and very labor and time intensive. And so using a single species or a group of species to kind of monitor what you're concerned about within that environment is a great, cheap, and reliable way to do it. And again, the environmental quality based on the species you choose is really important. So what makes a good indicator species, right? So you need to use the right tool. You need to use the right species or organism. If I was going to monitor those noxious gases in the mine, I probably wouldn't use that caddisfly. That's just not the right organism to use for that system. And likewise, if I was going to try to monitor the aquatic system or a river or a lake, I probably wouldn't use a canary. That's generally not the right species to use in those situations. So you have to really think about what species you want to use. And you also have to kind of focus on the concern. So what in the environment, what sort of component of the environmental health are you worried about or interested in? Is it some sort of pollutant? Is it some sort of disturbance, whether invasive grasses or some sort of anthropogenic disturbance or recreation going on that you want to look into? So you need to first target what you want to investigate in. And that kind of leads into your environment, right? So you need to decide where you're going to be looking at the environment and what kind of health system you want to investigate into. So would I be looking at aquatic systems? I'd probably be looking at species that live in that aquatic system. If I was going to look at terrestrial systems, I'd be focused on species maybe that live in the desert or in alpine environments. So you really have to target your species to the specific environment that you are concerned about. And then the biology of the species is really important as well. And I'll kind of touch on this a little bit later in my own research. But say, for example, you wanted to look at the collection or the accumulation of some sort of pollutant in the environment, say mercury, you'd want to select a species that encounters that mercury or that component regularly. And so you can provide some insight into that. And then spatial and temporal interests are also, an interest or a factor that you'd want to consider. And spatial and temporal are just fancy ways of saying the geographic range for spatial, and then temporal is just time change. So if you want to look at, out in the West Desert, say, for example, you want to look at specific areas, you might use golden eagle nestling, because those nestlings will have to stay in the nest for 10 to 12 weeks. And the adults will be bringing back food for those nestlings, but they're not going to be traveling very far, right? They need to be efficient with their energy as well. So that they'll be feeding those nestlings with food that is, within a close proximity to that nestling. So that provides you a good spatial kind of geographic range to look at. Likewise, you can also say, if I want to look at continent-wide accumulation of certain trace metals or trace elements, you could choose a group of different species that are spread out across the entire continent that also encounter that specific concern that you're interested in. So it doesn't necessarily need to be one species, and it doesn't necessarily need to be a small area. You can spread it across a very large area using multiple different species. And then the temporal interest, that time change thing, you can really look at, again, the eagle nestling example, you can see how, whether there's an increase in accumulation or a decrease in accumulation of, trace metals or trace elements or disease in these areas over time by testing them every year. You can test a nestling this year for those things, and again, test them next year and the following year. And that'll provide you a data point for each year and whether or not that concern, that environmental health concern, is present in that indicator species. So these are all kind of things that really you need to consider when you're deciding on indicator species, and it will be very helpful. So that's the end of part one, what an indicator species is. Does anybody have any questions at this point? All right, we'll keep going. All right, so the past and present use of raptors as indicator species. And just for those people wondering which species we have on the screen right now, we got an American kestrel up in the upper left. To the right of that is a red-tailed hawk. To the right of that is a Cooper's hawk. To the right of that is a peregrine falcon. And then we have a adult golden eagle feeding a nestling in the bottom left. All right, the history of contamination in raptors. The most classic case with raptors is DDT. And so I thought it was appropriate to use in this situation. And I'm sure a lot of people have heard it in passing, especially with the popularity of Rachel Carson's Silent Spring book. So let's talk about DDT a little bit. So it was developed in 1945 as a cheap and very effective insecticide. It was kind of primarily made so that by the U.S. government to prevent troops from getting malaria and typhoid and some of these very deadly diseases. And it's very, again, like I said, very cheap and very reliable and very effective. So they just started using it indiscriminately, kind of spraying it all over the place. And I'm sure lots of you guys have seen the photos of them spraying down kids, spraying it all over the beach, dropping it from the air. And at the time, they had done the research and thought there wasn't much of an issue. It has very low toxicity to mammals. We don't, in low doses, in moderate doses, it doesn't show any sort of harm to us. And that was kind of the end of the research. They weren't really interested in anything else. They didn't think this was going to cause much of a problem. And so that indiscriminate use just kept on going. Unfortunately, it is fat soluble, and it ended up bioaccumulating. And I'll talk about bioaccumulation in a minute, but first I want to kind of touch on Rachel Carson's Silent Spring book. So in 1962, she wrote a book called Silent Spring, where she basically raised the alarm and said, hey, we're dumping a lot of chemicals out in the environment. And not only are these chemicals harming ourselves, in large amounts, even DDT is very harmful to us, but it's also harming a lot of the wildlife that exists out there in the environment. And in doing the research for this presentation, it was really fascinating to hear the pushback she got. A lot of the male scientists at the time said, oh, you don't have an advanced degree, you don't know what you're talking about. And she got a lot of pushback. Even the USDA actually created what I would call maybe a propaganda video in the 60s to kind of discredit Rachel Carson to a certain extent. You can actually find it online. It's called Fire Ants on Trial. And it's essentially a video. It's only about 30 minutes. And in it, the USDA kind of talks about, oh, we have this scourge of this invasive species of fire ants. And the only way we could, they're damaging human interests and crops and livestock and just ruining people's lives. And through human ingenuity and the development of chemicals, we were able to take care of that scourge. And life is great again for all the people involved. But again, they just kind of gloss over any sort of wildlife impact. So it's really fascinating, and I definitely recommend folks go watch that if you're interested. And also read the book. It's fantastic. But in it, Silent Spring, she talks about how in the environment, and actually I believe it took place in my alma mater back in Michigan State, but she talks about how she no longer heard birds singing, and that that she theorized that this population of songbirds and other birds were declining. And she was right. And so what happens actually with DDT is it actually binds with calcium. And so it draws calcium from other locations to bind with it to make a new organic form. And when it does that, it actually draws some of the calcium out of the egg. So when those birds went to go lay eggs, the eggshells would actually be quite thin. And with that, when the adults would brood or incubate those eggs would often crack and no young would be born. So you can imagine if there's no young born, no input into the population, but you still have a death rate, the population is going to decline rapidly. Now, you're not going to have any inputs. and you're not going to be able to stabilize that population. And in fact, that's what we saw. So number of bald eagle pairs in the lower 48, you can see in 1972 is about when the ban in the US started, and they limited it from use. And you can see 1963, 487 pairs. And then after that, as we stopped using in the US, the number of pairs grew. And yep, in 2006, it was about 10,000 nesting pairs in the lower 48 bald eagles. And the same thing for peregrine falcons. They were a little bit slower to recover, and actually they had a lot of support. Tom Cade and the folks that started the Peregrine Fund were really instrumental in bringing these birds back from the brink. They did a lot of hacking, which is essentially just kind of raising the young in an enclosure somewhere else, and then releasing them into the wild to help propagate this population. And so I'll touch on the bioaccumulation bit here for a second, and I'll continue to kind of go back to it throughout the presentation. So if I'm not very clear with it now, feel free to ask questions later as well. But the way I'd like to think about bioaccumulation is a pyramid. So we have a pyramid, and this pyramid has 3 levels. At the very bottom of that pyramid, we have earthworms. In that second level, we have robins, American robins, a songbird. And then we have at the very top of that pyramid, we have peregrine falcons. So when DDT or any other contaminant, pollutant, anything like that gets pushed out into the environment, when we look out during the winter and see all that smog in, Salt Lake City or in the Wasatch Valley, all that stuff eventually settles and deposits down on the ground, or deposits on your car or on your lawn chairs and things like that. It eventually settles out. So as that DDT settled into the ground, the earthworms did their earthworm thing and ate up some of that DDT and absorbed some of it into their body as they ate some of that dirt. And then as the robins came along and ate those earthworms, they actually absorbed that DDT as well. So we'll say that every robin ate 10 earthworms and absorbed 10 earthworms worth of DDT. Well, come along the peregrine falcons then eat the robins, and we'll say every peregrine falcon eats 10 robins, and that peregrine falcon now has 100 earthworm quantities of DDT essentially. So it just continues to magnify up the food chain. So yeah, again, that pyramid, and as it's fat soluble, it just continues to grow and grow in the food chain until it gets to a level of very high within those apex predators. So that's kind of the classical example of using raptors as indicator species, seeing that there was something wrong. And here's kind of the current and the future of pollutants in the environment. And these things have been called legacy pollutants, because a lot of these things were actually made in like the 1940s and had been used in the environment for quite a long time. And they're probably not going to go away anytime soon. Certain things like the anticoagulant rodenticides, which is a big issue right now, and HawkWatch International is doing some work on that. That stuff has a half-life that can, degrade over time. But things like lead and mercury, those are elemental. Those will never go away. They may change forms into different organic or inorganic forms, but they'll never go away. The per and polyfluoroalkyl acids, those PFAs, those were developed in the 1940s, and they were made specifically not to degrade. They're water resistant, they're oil resistant, and so it's a real big concern about those things. And those things were put on everything from Teflon pans to food container wrappings. I read some study that said folks that even wax skis have high amounts of PFAs in their blood because they come in contact with chemicals that actually have PFAs in them. And they've actually said, most wildlife and most people have some amount of PFAs in their blood. And so some of the other ones that, are currently in focus around the world are veterinary pharmaceuticals. Obviously ranchers and agricultural farmers give pharmaceuticals to their animals, their investments, to make sure that they make it to slaughter and they can sell the products. Lead, obviously another elemental thing that is also used in a lot of ammo, in gunshot and also in fishing gear. So that's out there. Mercury, and especially methyl mercury. Methyl mercury is actually the more toxic version of mercury. And that's, that's out there and that's never going away. And both of those things are still pretty serious issues even here in the Great Salt Lake area. And so all of these are kind of the current and most important legacy pollutants, in my opinion, that are out there in the environment. And you can see there I have some sources of, just numbers basically. If anyone wants any sort of citations or any of my sources, feel free to contact me through email or afterwards. I'm happy to send you guys papers on all of these things. So the negative effects, right? So that's what we really care about. So if you develop, if you reach some certain threshold of lead or reach a certain threshold of mercury or these PFAs, you're going to have negative health effects. And like I said, these are also toxic to humans, and they also bioaccumulate. So you can tell that, those apex predators, those raptors, are going to be significantly impacted by these things. So what are some of the negative health effects? They impair reproduction, right? Embryotoxicity, those eggs just never hatch potentially, or they have developmental abnormalities when they do hatch. The cognitive function, all of these things really increase the chance for mortality or for predation. It's survival of the fittest out there. And if you're a raptor or an eagle born with some sort of abnormality, you're probably not going to survive for a very long time. So even when these things maybe don't get to a point where they're toxic enough to just outright kill something, they can also cause a lot of issues that potentially lead to an early death or lead to some sort of mortality that wouldn't have happened had they not had these pollutants in their environment. And just some other ones, physiological regulation, that clotting factor, and some hormone regulation that's also in there, especially with the anticoagulant rodenticides. And kind of some of the scary stuff is that some of these PFAs, especially, and some of these veterinary pharmaceuticals, there are hundreds of different chemicals out there that we still don't fully understand the potential that they can have on health effects within wildlife. So there's still quite a bit of unknown out there, and that's just emphasizes the point of using raptors as indicator species to help monitor some of these issues. So just some papers that have come out recently on the subject. This first one talks about the legacy persistent organic pollutants in the Great Lakes with common terns. The good quote from it that I pulled was, overall small chicks contain greater concentrations of pollutants than medium chicks and juveniles, likely from maternal loading to eggs. So there's two things there it's really important, right? So the higher concentration of pollutants, the smaller the chicks, right? The less likely they are to be fit and to be able to survive in the environment. That's really important. And then also this maternal loading into eggs, right? So just like DDT affected the eggs of the adult females when they were laying eggs back in the 60s and 70s, these chemicals can also be passed down into the chick. So it's not necessarily that the chick needs to be fed things that then affect their survival and their fitness. It could be that the adults actually have been affected or exposed to these chemicals and then pass those down to the young. And that can then affect their fitness as well. So very important stuff with that fitness component. And then this next one with Peregrine Falcons is an incredibly interesting paper. And essentially all it's saying is that we found higher PFA levels in urban nestlings than in rural nestlings. And that right there should worry us as well. Again, we use these birds and these species to provide an indication of what the health of the environment is like. And it also provides an indication of what our health might be like. If we're saying like, oh, there's a lot more chemicals in these urban environments, that should, send up a red flag that maybe we should be looking deeper into these issues to make sure our health isn't being impacted as well. So great papers. A couple more. Occurrence of veterinary pharmaceuticals and golden eagle nestlings. Scavenging is an avenue of exposure to chemicals. And that's with the golden eagle nestlings above, out of a study out of Spain. And then this lower one is with Benelli's eagles. And they looked at PCBs and DDT actually, and the effect it had on their metabolic and physiological health. And they showed that contaminated food resources affect nestling health. So not only does that maternal loading in the egg cause, potential negative health effects and detrimental effects to the fitness of those chicks, being fed food that has those pollutants in them also can affect that chick's development, right? And so during this developmental phase, it's incredibly important that we monitor these things. If there's even slight toxins in these things that can impact the development of these birds of prey. Again, both these are great papers as well. And before I end this section, I want to end on this photo real quick. This is a photo that was taken from one of my trail cameras out in the West Desert, and I'm over by the Nevada border. You can see this chick here. Excuse me. This chick here, it's May 17th, about 7 p.m. This chick is just looking out at the sunset. It looks incredibly healthy. There's food in the nest. We got at least 2 or 3 gopher snakes. Looks like we have a jackrabbit with quite a bit of flesh still on it. In the photos leading up to this, actually, this chick was eating. And at 7:30, the next photo I have of this chick, it has moved to the back of the cave and laid down. And that's very common behavior for these nestlings. They'll push back against the wall and go to sleep for a little while and get up and move around. Well, this chick moved to the back of the wall and then never woke back up. And it had died that day. And it's a complete mystery to me. This chick looks healthy. It was moving around. It has plenty of prey resource, a lot of food readily available for it. The adult attendance was great for this nestling. It wasn't predated. I have photos of all of it. But for some reason, this chick died. And this, to me, is the red flag. This is why I use, eagles as environmental indicators. What's going on in the environment that might cause it, otherwise healthy looking chick to die. And as we get into my research here in a second, we did take blood samples from this bird, and we'll hopefully have some understanding of the mortality here pretty soon. All right, before I dive into that, do we have any questions at this point from that second portion? All right, we'll keep on going then. All right, so how HawkWatch is utilizing avian apex predators as indicator species. So this really is focusing on my research. It's a collaborative research between HawkWatch International, Dugway Proving Ground in Utah State. And on the left, we have this great kind of example photo of a healthy looking nestling with a full crop. It's got a lot of food in its crop there, with a California gull and a gopher snake in front of it, right? So keep that in mind. And then on the right, we have a fascinating photo from one of my trail cameras at a nest. And the adult has a adult great blue heron brought back to the nest for prey for the nestling. And that photo is especially interesting because, one, it'd be terrifying to try to take down an adult great blue heron. But second of all, those birds are high in the trophic system, right? So they're also apex predators in that aquatic environment. And so things like mercury, other contaminants like selenium are bioaccumulating, increasing up through the food chain. So like as selenium or that mercury is depositing into the sediment of that aquatic system, bugs are then incorporating that into them. Small fish then eat those bugs, larger fish eat those bugs, and then great blue herons eat those fish. And that all along the way, you get an accumulation of those the chemicals. And so you have to wonder, again, during this developmental phase of these nestlings, it's a really important time when they're trying to develop their immune system, put on body mass for them that they can survive after they fledge from the nest. All these really important things. What potentially is in that great blue heron? What kind of contaminants, and what is that chick going to be exposed to? And just a side note, that adult golden eagle knew what it was doing, because in the next day it actually brought back the nestling great blue heron from that area. So it knew where to go for food resources. All right, so yep. So basically my research is kind of looking at environmental stressors and the impacts on nestling productivity and post-fledged survival. Again, how these things impact the development of the nestling and then survival afterwards. So, I mentioned Ken Keller just a second ago. Ken Keller is an amazing citizen scientist, biologist. He has done so much work with golden eagles in Utah for over 30 years. his data set is incredible, and he's been kind enough to let me look through it. And when I looked through the reports he had written, I found that about 70% or so of the diet of golden eagles that he monitored out in the West Desert were their prey base was mostly rabbits. So that 70% were rabbits. And so, we have an opportunity to place these cameras and actually monitor more of what these birds are eating to kind of get a bigger picture. He was doing passive sampling, so when he would rappel into the nests, he would look at prey remains, whatever bones and pieces were left over in the nest, and then toss them out, count them, and then come back later, the second time, do the same thing, and that's how he got his data. With these cameras, we should be able to get kind of real-time understanding of what they're bringing back. So if they bring back a jackrabbit, they eat the jackrabbit, and the adult then takes away that garbage, essentially, for lack of a better word. That might be missed in a passive sampling technique, but we'll hopefully be able to capture all of their diet composition with these trail cameras. So we'll be looking at the diet composition of these birds. And with that diet, we can actually then kind of piece out a baseline level for these toxins or these pollutants. So we're actually taking blood samples, which I'll talk about in a little bit, and we're doing a toxicology analysis on that to see this. So it should be really interesting, and this year was actually the first year we did it. So in 2020, we'll be able to tell, okay, this was the composition of their diet, this much was of rabbits, and this much was of snakes, and this much was of waterfowl and birds, and this is the level of lead, mercury, selenium, cadmium, arsenic. These are the baseline levels we saw from that baseline diet. And that baseline diet is really important because rabbit hemorrhagic disease 2 has now been documented in Utah. And it's a very unique opportunity that I have. It's unfortunate that RHD2, that rabbit hemorrhagic disease 2, has been documented in Utah, but we have a great opportunity to look at the diet shift, and then that diet shift will provide us some indication of the environmental contamination of these other, lead, mercury, other toxicants, and then see how that diet shift then incorporates into these nestlings. So a little bit about rabbit hemorrhagic disease 2. It has between an 80 and 90% mortality rate of rabbits. So that's pretty high. I mean, that's going to decimate the rabbit population. And it actually it's highly viral. It spread very quickly this year. I believe it was first documented sometime last December. And by July of this year, it was finally documented in Utah, kind of in the southeast and then in the central part of the state. So by the time my season had finished, it was kind of finally making its way in. So my first year, this year, of data collection is without the rabbit hemorrhagic disease. And then, so I'll have a good baseline. And then going forward, these next couple of years, when we see this kind of fallout from RHD2, we'll kind of see that change in diet composition, change in contaminant exposure. So how are we collecting this data? We're not, excuse me, we're not solely looking at contaminants and using these species as indicator species for contaminants. We're actually also using them as indicators for disease and parasites as well. So we draw blood from these nestlings, and I use a portion of the whole blood for my toxicology analysis and a portion of the whole blood for my excuse me, my blood parasite analysis. I'm looking at blood parasites and ectoparasites, kind of like ticks, and then like leukocytozoans, the things inside the blood. Those have been shown to have detrimental immune function effects on nestlings as well. So we're looking at those. We're looking at the toxicology analysis. And then we're actually centrifuging the blood down to get plasma. And then with that plasma, we'll be looking for antibodies of avian influenza, Newcastle disease, and West Nile virus. Again, all three things that are of importance for human beings. And then, so we'll have these blood samples and plasma samples to kind of look for those toxicants and antibodies of disease and those blood parasites. And I also, with the help of other HawkWatch International staff, but also Keller, was able to collect quite a few eggshells. So I believe I have around 32 different territories worth of eggshells spread across all of western Utah. So that's a really good data set, and that provides kind of a good blanket of data for across western Utah. And these eggshells are actually really interesting because you can analyze them, like I talked about before. There's this maternal loading. So the adult females will actually pass some of their toxins and some of their pollutants that they've accumulated, been exposed to, in through those eggs. And so those eggs actually contain some of those toxicants. And you can actually, what they'll do is they'll grind them up into a mixture and then use mass spectrometry to measure it, which very, very basic term. Please don't question me on this. But the very, very basic way is that they essentially boil it off or cook it off. And as the different elements then cook off and evaporate, they can measure where it comes out at, and then they can identify that chemical. It's very, very very basic understanding of the mass spectrometry. But basically, eggs are a great opportunity to study some of those toxicants as well, especially the PFAs. The PFAs are really kind of what I'm hoping to do with the eggshells, and I'm currently looking for funding for that. But I think PFAs are probably going to be the future of kind of, future interest for a lot of people. The EPA has actually put out, with a, bipartisan congressional mandate of $6 million, has put out this funding to study PFAs in rural areas because it doesn't break down. It is such a concern to human health and the potential accumulation in the environment for wildlife. So these eggs that we've collected should be really interesting pending further funding. And then we've also collected feathers from the nestlings. And with these feathers, we can do a DNA analysis and also check for other types of contaminants like lead and things like that. And I just want to also point out that this is all very non-invasive and non-destructive. We don't have to kill any birds to get any of this data. The amount of blood we're drawing is, very, very small, less than 1% of their body weight. And we actually offset it with a basic kind of like a saline solution so that they are hydrated after. We don't give them a cookie, but they do get some saline solution. So we do stuff for them. So that's how we're testing. So just a little bit more about the project as well. So we're looking at the diet, how that diet changes then affects that baseline of contaminants we're seeing. But we're also looking at the landscape variables. So do we see, like that Peregrine Falcon paper I'm a lot of the swine industry. So a lot of those pig farms down by Cedar City, west of Cedar City, by Lund and stuff. If they get a case of Newcastle disease in their pig farm or their operation, they usually have to call the entire operation. And if they were to do that, they could try to point the finger at the wildlife and say, hey, we didn't bring this in. Newcastle disease wasn't here. It had to have been from the wildlife. Well, we can say from our research, by testing, we can use these eagles as indicator species because they do encounter a lot of other species when they're hunting and things like that. We can say, hey, look, we've tested all these birds and we did not find Newcastle disease in any of these birds. So you shouldn't be blaming any of the wildlife for this disease. So it has a lot of kind of economic importance as well. And again, it's another indicator species for, these different types of diseases. So that's kind of the gist of my project, kind of at a basic level. It's a little bit more advanced than that, but I think for time's sake, that's kind of the gist of it. So how does it link up to immune function? How does this link up to post-flight survival? Again, like I talked about with that developmental phase, right? So as these nestlings develop, a lot of their energy inputs first go towards their immune function, right? Their chicks, as they're developing, they want to be able to fight off any sort of diseases or anything that's going to make them sick first, so that doesn't kill them, and then they'll invest the rest of the energy into, say, creating body mass, developing feathers, more of these physiological components within them. So looking at potentially what impacts the immune function of these nestlings will be really important to understanding, mortality, potentially how fit they are. We'll use the parasite count data, this disease antibody prevalence data, and the contaminant data to say, okay, we've seen this nestling had this much parasites, it was exposed to these diseases, and it has this much contaminants in it, and we'll be able to say, okay, cool, it's a baseline for this bird. And then we also, to a sub-selection of these nestlings that we're working with, we actually attach satellite transmitters. And with these satellite transmitters, we can actually track their movements after they fledge. And that will actually provide us an indication of, okay, we saw, all of these different levels of contaminants or disease or parasites within these birds, and this is how long they survived post-fledge. Do we see any trends in, contaminant level? Do birds that have this, a certain threshold of lead or a certain threshold of mercury, do they survive longer than birds that have a lower threshold than that? The birds that have lower amounts of those contaminants. Do we see birds that have antibodies for West Nile virus or avian influenza survive longer? Because now they have the antibodies for the diseases and are no longer maybe susceptible right away to those diseases once they fledge from the nest. So in conjunction with this understanding of immune function and how these components impact that immune function, and then kind of how that then influences their post-flight survival, we'll really be able to fill a lot of knowledge gaps on how these nestlings develop, and then what potential causes of mortality occur to them in the wild. And hopefully, with this information, we'll be able to kind of get a better sense of the population and how these nestlings, these early individuals, can then add to the overall population. And just actually another, changing gears a little bit, this is another developing project that I've been talking with a collaborator, another PhD student up at Utah State, and I think is particularly relevant for those of us that live in the Wasatch Valley. So, when the Industrial Revolution started, around the, in the 1800s till about 1950 or so, there was a lot of mining, a lot of manufacturing, obviously, a lot of refining going on of different things. And there were really no environmental laws. And so a lot of this pollution was being dumped into the atmosphere. And like I mentioned earlier, it deposits. It doesn't stay in the atmosphere forever. It slowly settles down into the ground, into the soils. And here in Utah, a lot of it settled into the Great Salt Lake. Before we had built that causeway, when it still had quite a bit of water in that Great Salt Lake. And so when it settled into the Great Salt Lake, those toxins, those contaminants settled in, they all floated down to the bottom of the lake, and then got kind of ingrained into the sediment, and so year after year after year, more contaminants kind of floated down into that lake bed and just settled in. So when we built a causeway and kind of dried out that northern half of the Great Salt Lake, and as the water levels have fluctuated in the Great Salt Lake over the past 50 years, a lot of that lake bed has become exposed. It has become a playa, and as that sediment gets exposed, it can get kicked up by the wind. And so as that wind kicks up that dust, well, that dust actually also contains pieces of those chemicals and those trace metals and those trace elements. And it kicks it up into the air, and so you have this kind of toxic dust kind of blowing around. And great research, absolutely fantastic research out of BYU and out of Utah State has been occurring, looking at kind of what's been going on with that. And there's particularly a PhD student that I'm working with that is assessing the bioavailability of those trace elements in the dust and trying to determine where their deposition is along the Wasatch Front. And so she's able to say, okay, where is the dust going? What's in the dust? What's attached to that dust? and how mobile are certain elements, and could they be for organisms or in the soil. So basically, she can put out these fancy sensors that say, okay, this is how much dust is occurring in the area at this location. These are the elements that are attached to this dust, and this is what's potentially available in the environment. And that's where I see myself and HawkWatch International coming in, because then we can test those raptors. We can use those raptors as an indicator species to say, hey, that dust that's being blown in, we're really not seeing it bioaccumulate in the ecosystem. We're using these apex predators as indicator species, but we're not seeing it. Or we can say, yes, those exact trace elements that you are looking at that are found in the dust in these areas, yes, we actually are seeing those elements in high quantities in these apex predators, in these indicator species of raptors. And so this is kind of a developing project, and hopefully we'll have some more kind of information on it coming later this winter, and be able to put into effect next year. And so that's just kind of a side project that we have going at the moment. So with that, I would like to thank all of my supporters. Robbie Knight at Dugway Proving Ground has been absolutely instrumental in helping support this project. Absolutely could not do this without his funding. It's been crucial. Steve Slater at HawkWatch International has also been a fantastic mentor and has been great at kind of advising me through this process and helping me kind of advance to this point. And then also really want to thank Dr. Clark Rushing up at Utah State for taking me into his lab and providing me some of that academic mentorship as well. I really couldn't do this project or any of these things without these folks' support. So I want to thank them a lot. And with that, I'll take questions. All right, looks like we have some questions in the chat. Is the chick on the right banded? I can't tell you which chick it was, but basically, yes, if all the photos from the trail cameras were banded, yep. So we put bands on all the nestlings that we sampled as well, so that hopefully we can get those kind of an understanding of where those birds are going, or if their carcasses are recovered, we'll know where they went, and potentially, hopefully, the reason for mortality. Yeah. So blood taken in May, no results yet. Yep. So we're working actually with the state of Utah Veterinary Diagnostic Lab, and they are very, very kind to me and let me use all their lab facilities and their centrifuges and whatnot. And so the toxicology analysis is ongoing. I think we have samples from 40. I think we have like 47 different blood samples. A lot of these chicks we sampled at about 3 to 4 weeks of age, hoping to kind of gain some of that understanding of that maternal loading. Do we see any sort of antibodies from diseases or toxins from potentially the adult? And then we test them at 7.5 weeks prior to fledge to then get an idea of how, what has the diet contributed to their development of these contaminants and diseases and stuff. So we have a better idea of that. But hopefully, yes, hopefully by the end of the winter, we'll have all these toxicology analysis and other results back. So any data from feathers as well. Yeah, so feathers are, excuse me. Feathers are an interesting thing. There's more and more technology coming out now where you can actually use the feathers to analyze them for, say, lead and other toxicants. At this point in time, I think with the funding that we have, we'll probably save them for those DNA kind of sequencing stuff. But as technology becomes cheaper, we might be able to use them and utilize them for other things. Yeah, so have I, Great Nest camera footage, have you considered sampling the prey base for contaminants? So this kind of goes back to my explanation of using indicator species, right? So it would probably be pretty time intensive and very costly to go out and sample all the snakes, to do a very statistically rigorous sampling of snakes across the West Desert and in these territories. So basically by using the trail cameras to capture their diet composition, and then extrapolating kind of the toxicant levels and the factors we're looking at, really helps us kind of eliminate a lot of that labor and time and cost-intensive efforts. Let's see. How long will the study go on for material conclusions to be made? So this is currently my PhD work right now, and so we'll be doing 3 years of field work at this point. And so we just finished our first season, and so by 2022 we'll have finished all of the field work, and I'll be hopefully be able to write up my chapters and get some things published after that in that 4th year. I'm trying to shoot for that 4th year ending. But I can easily make available kind of interim updates, and I'm sure we'll post those on HawkWatch International's website for folks to read and get updates on the project. What is your sample size for the blood work? That's a great question. So the sample size, this year we sampled 27 different nestlings. I think that was 22 different territories across western Utah. And the study area essentially covers from the Idaho border all the way down to Arizona. So I have nests that are outside of St. George all the way up to by Lucin Spring, areas like that. So we're kind of covering a very large latitudinal gradient. And the kind of goal for that is to get a good sample of the West Desert and kind of see what's going out in those areas. And like I said, we also did sample twice, once at that 3 to 4 week age to see about that maternal loading and see what's going on early, and then again right before fledging. So why are the lab results taking so long? Yeah, that's a great question. No, it's primarily because the state lab is cutting us a break with a lot of these costs and allowing us to do a lot of work out of the lab, and so our samples are kind of not put on the back burner, but are analyzed as time allows for them. And I'm not particularly concerned. I know I'll get the results by this winter, so I'm not particularly concerned at this point in time. I'll have plenty of time to kind of pivot or change some methods of my study in the spring before I go back out to do some more sampling come next April, May, June, July. So yes, it would be nice to know kind of why that golden eagle chick died that looked so healthy in May, but we'll find out eventually. Right, aquatic prey is more likely to contain high rates of chemicals than other prey. So that's a great question. We've seen, there's some papers out there. There hasn't actually been a lot of research done on reptiles and contaminant. They are higher on the food chain, so you would expect that they would have similar bioaccumulation issues as other species that are higher in the trophic levels or higher in that food chain. But there hasn't really been a lot done on snakes. And so I'm thinking that they have shown that there is cadmium, and there potentially is maybe some selenium in snakes that are near water bodies, but hasn't really been done a lot of work on that yet. And as far as high rates of chemicals and other prey for aquatic resources, absolutely. So here in Utah on the Great Salt Lake, there are actually waterfowl advisories. So I think, you're gonna put me on the spot here. The goldeneye and one of the teal species. There's actually, so you can't eat more than 8 ounces of their meat in 1 month, because they accumulate such high amounts of methyl mercury that it's bad. It's very similar to the reason you can't eat, trout every day or salmon or walleye. These fish that are kind of sought after by anglers. They have such high rates from that bioaccumulation or that kind of, the increase every step of the food chain, that there are those advisories. So yes, there are definitely high rates of chemicals in them. How are American kestrels doing? That would be a question for Dave Olier or Jesse Watson. Those are the American kestrel specialists, and I'm sure they'd be happy to answer any and all of your questions. And I think I'll leave it at that. In your early analysis, have you found more than trace levels of blood lead in your nestlings? So again, the toxicology analysis hasn't come back yet. I know there's been a lot of work done on lead levels out in California with Chris Venom. And he saw that trade-off between, well, they have a lot of ground squirrels that are being shot so they can provide a lot of biomass or food for these nestlings, but at the same time they're having these physiological impacts from the lead that are maybe counteracting it. And it's a really fascinating paper out there. Chris Venom's one of the authors on it, and he's done some great work on that stuff. And I know there's been a lot of other lead things, and hopefully once we get back the analyses, we'll have a better idea. But if I was to make a guess, I think we'll probably end up seeing a lot of lead probably in our more urban, the nestlings and the nests and the territories that are close into more urban environments. Then we do say we'll be out more in the rural areas and, far out in the remote West Desert. And again, that's part of the study, is to see if we have changes in these chemicals, the toxicants, these diseases, these parasite levels based on the diet and the landscape variables. And how much blood you collect from each bird. So again, we are limited to less than 1% of their body mass, and we generally take way less than that. We're really just shooting for, when we're doing like 3 to 4 week old chicks, maybe 1 cc of blood, maybe 2 it depends on the weight. If it's a really healthy nestling, then we'll maybe take 2 cc's if they're above, 2 kilos or so. But again, it's a very small amount. And then usually from the 7 to 7.5 week old birds, we can get a little bit more, maybe say 3 cc's or so of blood from them. So we can do a little bit more of an analysis on the older chicks, which is helpful because they're just about to fledge. And then we'll have a better idea using those PTT, those satellite transmitters, to kind of understand what their health is like, their immune function is like pre-fledge, and then see what their movements are like once they leave the nest. Right. Any other questions? All right, great. Well, I really want to thank everybody for attending the talk today. And, I would just want to throw out that, your support really makes HawkWatch International run and really supports a lot of this research. So if you feel like donating, feel free to contact or go to hawkwatch.org/support. And, whether it's a financial donation or even volunteering some time, we'd be happy to have all the support we can get, especially in these times. So again, thank you all for attending my talk, and feel free to reach out to me if you have any further questions or you would like to talk to me about any of the papers that I cited or the chemicals I discussed. I'm always happy to chat with folks. All right, well, thanks so much, guys.